The routine procedures followed by surgeons before and during operations are designed to erect barriers against infection and disease caused by microbes. Today we recognize many thousands of species of microbes. By using motion pictures and a special microscope, bacteria can be observed as they multiply. Relatively few species cause disease, and these are called pathogens. The great epidemics and plagues that time and again devastated cities, countries, and even continents were caused by pathogens. The first man to see bacteria and other microbes was the Dutchman Van Leeuwenhoek, who built a practical microscope during the middle of the 17th century. This primitive instrument had a single biconvex lens and an ingenious device for focusing. Van Leeuwenhoek obtained such high magnification with this microscope that he could observe small animals in detail and discover bacteria. In 1683, he published drawings of his discoveries and included the bacteria. At one time, smallpox was epidemic throughout the world. About 100 years after the discovery of bacteria, Edward Jenner, an English country doctor, investigated the belief that farmers and milkmaids who had cowpox did not later contract smallpox. We now know that cowpox is a harmless disease closely related to smallpox. He removed a sample of pus from a pustule on the hand of an infected milkmaid and inoculated the pus in the boy's arm. Jenner deliberately infected a healthy 8-year-old boy with cowpox. After 6 weeks, the boy was again inoculated, but this time, pus from a vesicle from a patient with smallpox was used. The boy did not contract smallpox. This simple experiment was the beginning of vaccination. The discovery of smallpox vaccination was first greeted with skepticism and then with ridicule. Vaccination became the target for many jokes and cartoons. In time, vaccination centers were opened all over Europe, and then the rest of the world. Once a major menace, smallpox was eliminated from those countries that enforced vaccination. As microscopes improved, it was possible to demonstrate microbes in the fluids of patients with an infectious disease. One simple question, however, could not be answered. Were these microbes the cause or the result of the disease? The correct answer was provided by Louis Pasteur, a French chemist who in 1856 was studying the fermentation of beet juice. Pasteur examined the fermenting juice with a microscope and saw multiplying microbes, mostly of a type called yeast. In other fermentations, he observed various types of multiplying microbes. According to the then current theory, microbes were the product and not the cause of fermentation. Pasteur believed that the multiplying microbes were responsible for fermentation. He boiled samples of beet and other juices in flasks to destroy any microbes that might have been present. Some flasks were immediately sealed to protect the juice against contamination from the air. Only the sealed flasks did not turn cloudy. The open flasks had multiplying microbes. Critics of these experiments maintained that the boiling and sealing had prevented fermentation by eliminating oxygen from the juices. Pasteur repeated the experiments, but added a long and twisted neck to each flask so that oxygen could reach the juices. Specks of dust or microbes were trapped in the bends of the twisted neck. This time, only the flasks with twisted necks remained clear. Pasteur had answered his critics with a simple experiment. In 1865, Pasteur was asked to investigate the silkworm diseases which were ruining the French silk industry. He went to LS, an important center for raising silkworms, and started to work with all the detailed methods developed during his study of fermentation. Pasteur crushed the bodies of worms and moths with the pebrine disease and found a particular type of microbe on microscopic examination. Using suitable tests, he proved that this microbe was responsible for pebrine. This investigation provided the first demonstration that a specific microbe could cause a specific disease in an animal. The study of microbes, now called microbiology, was greatly advanced by the work of Robert Koch, a German doctor who practiced medicine in a small Silesian town. He built a laboratory in his home at his own expense and developed many techniques to conduct his research. Working alone in this laboratory in 1876, he investigated a common disease of livestock, anthrax. A rod-shaped bacterium had been thought for some years to be responsible for this disease. Koch isolated the bacterium from diseased animals and then proved that the many forms, including the spore, were all members of the same species, Bacillus anthracis. Furthermore, he demonstrated how this pathogen was transmitted to healthy livestock. Koch was a very careful worker, and many of his methods are still in use. Microbes had previously been grown in a broth. Therefore, it was difficult to get pure culture. He introduced the solid culture medium by adding agar to the broth. Agar is a product from seaweed that hardens at room temperature after cooling. The bacteria produce separated colonies on the agar medium, which can be used to obtain pure cultures. He also developed a simple method for observing bacteria. The bacterial colony was smeared on a glass slide, killed, and fixed by gentle flaming, and then colored with a dye or stain. Koch was the first to photograph microbes through the microscope. And his photographs still show high quality. Using all these techniques, Koch discovered the tuberculosis bacteria and demonstrated the microbes in the tissue and sputum of patients. What are microbes? They are single cells that multiply rapidly by dividing to produce the cloudy broth or colonies on solid mediums. The growth of a colony of a soil microbe can be shown with motion pictures. The rapid growth can be appreciated by noting the passing of time on the clock in the corner of the pictures. Some microbes are motile. In the pictures, the spirochetes are rapidly moving among the red blood cells from an infected bird. The rod-shaped microbe, Bacillus subtilis, and the bacterium causing cholera move by means of a whip-like flagellum. Other microbes, such as Staphylococcus, Streptococcus, and the tuberculosis bacterium, are not motile. To get a clear idea of the size of microbes, we must journey into the infinitely small. The sewing needle in the picture is 3 centimeters, or a little more than 1 inch long. The eye is 25 1/100ths of a millimeter, or about 1/100th of an inch. The nylon thread through the needle's eye has a diameter of 50 microns, or 1/500th of an inch. Next to the thread, which now seems enormous, are treponema, a microbe approximately 7 to 10 microns long. An electron microscope is needed to see details. The Salmonella bacterium is 2 to 3 microns long, and attacking the bacteria are bacterial viruses or bacteriophage. Of more recent discovery, the viruses are assuming greater significance to man. Some viruses are pathogens for man and animals. For example, smallpox, influenza, and poliomyelitis are caused by viruses. How can such minute organisms endanger health? Bacteria and viruses attack the host in at least 2 ways. First, they multiply and destroy various tissues and organs of the host. Second, they may excrete poisons called toxins in the host. Fortunately, the host has defense mechanisms. Here we see a drop of blood in which have been placed 2 colonies of bacteria. The white blood cells rush from all directions to attack and engulf the bacteria. This phenomenon was discovered by the Russian bacteriologist Mechnikov, who termed it phagocytosis. We can see how the white blood cells, or phagocytes, push aside the red blood cells to surround, engulf, and digest the bacteria. Once the bacteria have been destroyed, the phagocytes disperse in the bloodstream. A second natural defense mechanism is the production of antibodies, a particular type of protein, by the host. The invasion of the host by microbes stimulates the host to produce antibodies which neutralize or destroy the microbial species that provides the stimulus. Once produced, the antibodies remain in the bloodstream for varying periods of time. If the same microbial species should again invade the host, the antibodies would provide protection. This phenomenon is termed active immunity. In 1877, Pasteur attempted to produce an immunity against the very destructive disease, chicken cholera, which he was then investigating. He grew pure cultures of the microbe in broth. When the broth turned cloudy, he started a fresh culture from the preceding one. To test the virulence of these serial transfers, he inoculated chickens. In every test, the chickens always died of cholera. In the summer of 1879, the laboratory was closed for the annual vacation of 2 weeks. To start the new transfers, the pre-vacation culture was used. When the new cultures were tested for virulence, the chickens showed some symptoms of the disease, but soon recovered. The surviving chickens were inoculated with virulent cholera microbes, but did not contract the disease. In other words, the chickens had been protected by a vaccination. Although the aged cultures could not produce a fatal disease, the microbes could stimulate the formation of protective antibodies by the host. Pasteur had discovered a method for making vaccines. In these scenes, we see the steps involved in the production of a typhoid vaccine at the Pasteur Institute in Paris. Vaccines are now available: typhus, cholera, yellow fever, tuberculosis, and poliomyelitis. Antibodies are not produced by the host in time to be effective against invading microbes. Is it possible to control infectious disease by injecting the patient with prefabricated antibodies from another patient who recovered from the same disease? In France, Roux filtered a culture of diphtheria bacteria and demonstrated a toxin in the filtrate. In Germany, Behring was able to inject animals with a toxin without killing them. He collected the blood from such animals, and after coagulation, the serum was found to contain protective antibodies. Sick animals could be saved and cured by injecting this serum. The phenomenon has been termed passive or acquired immunity. Roux eventually developed methods for preparing protective serums for a number of diseases. The horse is the best animal for producing antitoxin. After repeated injections with the toxin, several liters of blood are collected and allowed to coagulate. The serum is rich in antitoxin. After processing the serum, it is safe for injection into patients. Serum therapy has been very successful in controlling diphtheria and tetanus. While Pasteur and Koch were creating medical bacteriology, surgery continued to pay heavy tribute to infection. Surgical methods had remained unchanged for centuries. Few operations escaped serious infection. In amputations, for example, the death rate was approximately 60%. Lister, an English surgeon, knew of Pasteur's work in fermentation and believed that microbes might be the cause of surgical infection. In 1865, he used phenol in performing the first operation with an antiseptic. A solution of phenol was sprayed around the patient's body near the site of operation. The surgeon's hands and instruments were carefully disinfected, and the bandages and dressings soaked in phenol. By these and other methods perfected by Lister, surgeons were able to reduce the death rate to 15%. Antiseptics irritated the tissues and retarded healing. By following Pasteur's recommendation, the aseptic method was introduced into surgery. The possibility of contact between microbes and wound was avoided by strict cleanliness in the operating room and sterilizing all instruments and dressings with heat. commonly used in surgery. Continued research on antiseptics provided many useful compounds to disinfect the skin and small wounds. Unfortunately, these compounds were too toxic to be swallowed or injected. In 1932, Domar, a German chemist, discovered that a colored compound called quantacil could be safely injected and could protect against streptococcus infection. Soon after, French chemists isolated the colorless active component, sulfonamide, from quantacil. The sulfa drugs have been effective against many pathogens when applied to wounds, ingested or injected. At the time when sulfa drugs were being successfully used, the antibiotics were about to prove their value in controlling bacterial pathogens. In 1928, an English bacteriologist, Fleming, was working on staphylococcus in St. Mary's Hospital in London. One day, he noticed a fungal colony on a culture plate. Although fungal contamination was common, he observed that the colonies of staphylococcus had been destroyed only near the fungus. He was able to show that the fungus was releasing an antibacterial compound into the medium. Fleming named this compound penicillin, after the fungus Penicillium notatum. Fleming tested the potency of penicillin by a method still in use. A dish is made in the agar and filled with a solution of penicillin. Different bacterial pathogens are streaked at right angles to the dish. The inoculated plate is incubated for a few days and then observed. Sensitive pathogens do not grow near the dish. Injecting high concentrations of penicillin into laboratory animals had no ill effects. Although Fleming predicted the therapeutic value of penicillin, his laboratory was not equipped to exploit his discovery. 12 years later, Florey and Chain resumed the study of penicillin at Oxford. These pictures were taken by Florey and illustrate the original equipment used to grow the fungus on a large scale and to obtain large amounts of penicillin from the culture broth. They were able to produce a high-potency, non-toxic, stable antibiotic. Later research with fungi and bacteria from the soil has yielded new antibiotics. Soil samples are diluted and spread on the surface of the agar already covered with different bacterial species. Colonies with an antibiotic effect are isolated in pure culture for further study and testing. From such research have come streptomycin, aureomycin, and chloromycin. Many bacterial pathogens are now controlled by antibiotics. The fight against the microbes, begun more than 100 years ago by Pasteur and Koch, has conquered most of the infectious diseases that once decimated mankind. This fight, plus better living conditions and better food, have diminished the death rate for children and added years to the average life expectancy. The fight is not yet ended. In tropical climates and underdeveloped countries, infectious diseases continue to rage. Certain diseases are not yet understood satisfactorily and are probably caused by viruses. Research on infectious diseases is increasingly active. Better equipment and closer international cooperation are penetrating deeper into the mysterious world of the virus. These extremely minute organisms are located on the borderline between the living and non-living. Their study is important to understanding the nature of life.